All right, I believe that we're going ahead and getting started. My name is Dr. Jonathan Woodhouse. I'm the current president of the Connecticut Psychological Association's Neuropsychology Division, and we want to welcome you to the CPA CNS Virtual Neuropsychology Conference. We could not be more excited about this event. Special thanks to Dr. Sarah Raskin for putting together such an exceptional panel of speakers, as well as Drs. Tim Bellevue and Brett Steinberg, who worked really hard behind the scenes to pull this event together. In 2019, as many of you know, the new CPT codes were released, and neuropsychologists across the state immediately started to notice a drop in their reimbursement. Now, as many of you will know, Karen Postal et al. Back in about 2014 proposed what's known as the 360-degree model of advocacy that seeks to coordinate resources from the interorganizational practice committee to the national organizations like NAN, AACN, APA, to regional neuropsychology organizations, state psychological associations, and the clinician. Incidentally, I'm on the IOPC currently, and I'm in the chair of NAN's Professional Affair Committee. So when this issue hit, I was in a position to see how this played out at each different level. And I put the IOPC 360-degree model into action. I notified IOPC, APA, and they were ready to move. But it was actually CPA's lobbyist, Anita Shepker, who identified a computer glitch in the DSS system in Connecticut and was able to quickly work with DSS to get that issue resolved. It was such a swift and decisive victory for the division and really served to highlight the importance of being organized and connected to the state association's legislative advocacy apparatus. Then COVID hit, and everyone was forced to reinvent how services are delivered, and it accelerated the conversation on teleneuropsychology. As you may know, initially 96116 was the only code insurance would cover, and it took vigorous advocacy to get the other neuropsych testing codes covered during this public health crisis. The national organizations were focused, focused on nationally administered plans like Aetna, Beacon, Optum, and so on, but it was the Connecticut Psych Association that advocated for state-administered plans like Connecticut, Anthem, and Husky. We were successful in getting the codes covered, and when those extensions ran out, got those codes extended, got the coverage extended. And so these two events really crystallized the critical need for us as neuropsychologists to be connected, to be informed, and to be a part of what CPA is doing, advocating for our own professional interests at the state level. Moving forward, there are big issues on the docket: telehealth, diversity issues, research and training, and reimbursement. At this very moment, you may have heard that CMS is looking to reduce payments by 10.6% starting in 2021. Diane Howe said it perfectly when she said, quote, "Constant vigilance is required in order to maintain the status quo. Even if we like things the way they are, we have to fight to keep them that way, and to even improve the practicing climate requires our participation." And so none of this would be possible without you. If you're not a member of CPA, I do hope you will consider joining us for this important work. With that, I want to turn it over to Dr. Raskin, introducing our speakers. Thank you.
Thank you, John. I also just want to welcome everybody, and talk about how much I'm looking forward to this amazing lineup of speakers joining us here today. I also want to give a big thank you to the whole committee, Dr. Steinberg, Woodhouse, and Bellevue. It was a pleasure to work with them all to put this together. If you're interested in learning more about the Connecticut Neuropsychological Society, I welcome you to visit our website. I'll put a link in the chat box so that you can find it. There's information there on prior conferences, and we'll be hosting the tapes of today's event there. In the past, we've had bi-monthly meetings with both research and clinical talks or discussions over dinner. Our membership has reduced over the past few years, and I know we all welcome your suggestions for programs and ideas that might be of interest to you in the future. But we're particularly excited about this partnership with Connecticut Psychological Association and all of the advocacy work that they do, some of which you just heard about, and we very much look forward to more shared events. But most of all, I can't wait to hear from our speakers. So I'd like to start by introducing Dr. Tottenham. So welcome.
Thank you so much. Good afternoon, everyone. It's good to be here with you, and I just want to thank the association for this invitation. And so I'm going to share some slides with you. Okay. So the research that our laboratory addresses is a very old question in psychology, and one that many of you may be addressing in your own work, which is really, how is it that we explain this enduring link between early experiences that we have, both positive and negative experiences, and our later emotional functioning, in particular, emotion regulation, the ability to control or manage big emotions that we have. And the approach that we take is to study how early experiences are influencing the construction of the brain. And for the purposes of today's talk, I'm going to be focusing on two regions. One is the amygdala, this set of nuclei deep in the brain that are responsible for our emotional attention and vigilance, as well as learning about the relative safety and danger of the environment. Now, in adulthood, the amygdala has strong bidirectional connections with the medial prefrontal cortex, which is really well positioned to gather information from several areas of the brain, including memory regions, perceptual regions, other higher cognitive processes that by and large together help downregulate the over-arousal associated with the amygdala. We study how this process is unfolding during development. And the reason is because sometimes you find out answers when you're looking in the developing creature about how certain paths are likely to be predicted as compared to if you were to just look in the adult state. So if we look at a number of developmental disorders that are associated with difficulty in emotion regulation broadly defined, often you see figures like this, where the peak age of clinical onset is right at the beginning of the adolescent period. And so our approach is to not just focus on that early adolescent period to understand the developmental origins of these individual differences, but instead to also pay close attention to the antecedent period. So what's happening during childhood that is leading to differential construction of these emotion regulation circuits. So I'd like to share with you an early study that we had done in our lab, and this was when Dylan G., who's now at Yale University, she's a fabulous independent scientist on her own, but she was a graduate student in my lab, and we had invited 4 to 22-year-old healthy, typically developing individuals to come into the lab, and we presented them with fear faces. And the reason we chose fear faces is because of this wide age range that we had. Fear faces are a relatively innocuous way to activate amygdala activity in participants as young as 4 years old. And so with this approach, we could start to develop a growth chart, so to speak, across childhood, adolescence, and young adulthood of amygdala reactivity to emotional stimuli. And what we found was, in response to these fear faces, the amygdala shown here, the right amygdala, showed large age-related change. And that's what I'm showing you here. It's a little bit hard to see in this blob format, so I'll pull these data out to show you better here. That here's age on the x-axis, here's amygdala bold signal or response on the y-axis, and each dot is a participant. And what you can see is the strongest, most robust responses are occurring at the youngest ages. So we get this strong early amygdala response, and that made a good deal of sense to us because when you're younger and relatively new to this planet, you have a lot more learning to do about the relative safety and danger of your environment. So you want the amygdala kind of chugging along at early stages. So then we can ask about the amygdala's connections with the medial prefrontal cortex. And there are a number of ways to do this. The metric that we rely on primarily is called functional connectivity, which many of you may be aware of, but if you're not, functional connectivity works like this. You can pull the time course data out of one region like the amygdala and plot it here, and the time course activity of the medial prefrontal cortex. And the degree to which these two regions are correlated with each other or are in sync gives us an index of that functional connectivity. Now, in healthy adults who are well regulated emotionally, what you often find in response to things like fear faces is that the amygdala and prefrontal cortex show a high degree of connectivity, except that the connectivity is inverse. It's a negative correlation between these two regions, which is understood to mean that when the prefrontal cortex increases its activity, the amygdala decreases its activity. So that's sort of the neural signature that we expect to see in healthy, well-regulated adults, is this negative connectivity. So we use this metric in our participants from 4 to 22 years old. And again, we find this strong age-related change in connectivity, which I'm showing you here. I'm not showing you activity anymore. This is now connectivity between the medial prefrontal cortex and the amygdala. And to better visualize what the nature of this change looks like, I'll plot here. These are the young adults where we see that negative connectivity, that regulatory pattern in the young adults that I just described. If we look at younger and older teens, we still see that negative connectivity, but you can appreciate that the magnitude has now decreased relative to what we saw in adults. And that's consistent with what we know about the slow development of emotion regulation abilities. But what captured our attention most is what we observed in children, 4 to 9-year-olds. Here, we didn't see evidence of that negative connectivity, but instead we saw positive connectivity. That is to say that the nature of the conversation between prefrontal cortex and amygdala was different in childhood and then seemed to show this switch once we started getting into adolescence. Importantly, this switch in connectivity valence was associated with age-related changes in normative emotional behaviors. In this instance, it was looking at normative separation anxiety, which was changes in normative separation anxiety were explained by this neurobiological switch. So taken together, these data are indicating, if this is age on the x-axis here, that at least during childhood, if not earlier, we get a nice, strong, early amygdala response to stimulation in the environment. But that's happening in the absence of these adult-like connections with medial prefrontal cortex, which we don't see emerging until the beginning of adolescence. So that makes us very interested in this childhood period, when the nature of the neurobiology underlying emotion regulation abilities is so different compared to points thereafter. And might this be a good candidate time in life to examine 4 potential sensitive periods for this neurobiology, moments when the environment may have a particularly impactful effect on the way that the system continues to construct itself. Moreover, for reasons that I'll explain later, we have hypothesized that this strong amygdala reactivity in childhood may actually be the biological prerequisite for the instantiation of these top-down regulatory connections from prefrontal cortex that are ultimately going to quiet down the activity of the amygdala. So our working interpretation of developmental change looks like this. It may be that given the hierarchical nature of brain development, the fact that the amygdala is a much earlier developing region than the prefrontal cortex, that in childhood, the amygdala is driving a lot of the conversation to medial prefrontal cortex. And I often liken the amygdala to the older sister that got there first and learns about the world and can instruct the prefrontal cortex about what she's been learning. So that over the course of development, that helps instantiate these reciprocal top-down regulatory connections from prefrontal cortex that we see in adulthood. Now, that's just our interpretation of our correlational fMRI data, but there are a number of tracing studies in rodents that can, with more certainty, talk about this causal relationship, where you see amygdala sprouts to prefrontal cortex emerging developmentally earlier than the reciprocal top-down regulatory connections. So these patterns of development really remind us of a 200-year-old quote that the child is the father of the man. So in other words, when we just have an adult vantage point about this neurobiology, we often think about the cortex as controlling these subcortical emotional systems. But perhaps if we can appreciate a developmental view, we may instead view the amygdala as being a tutor, so to speak, of the prefrontal cortex. So to better or more formally address this question of whether the nature of connectivity at time 1 is actually predicting something about the nature of connectivity later on in life, we took our data that assessed that fear face elicited connectivity, or that stimulus elicited connectivity, in our participants from 4 to 22 years old. But we also collected a second modality, a second type of scan from these same participants called resting state, which many of you are familiar with, but if you're not, resting state connectivity is the same connectivity metric that I just described, except that you're collecting it while people are doing nothing. So essentially what you get is sort of a functional skeleton of the brain. You get a sense of who is hanging out with whom when the brain is not tasked with any particular job. And it turns out in adulthood that that resting state functional connectivity is highly predictive of individual differences in emotion regulation behaviors in adulthood. So we collected both modalities at time 1 across all of our different aged participants, and then sent everyone home. They grew up for 2 years, and then we brought them back and collected both modalities again. So one thing that we found was that age-related changes in both that resting state connectivity and the connectivity elicited by emotional stimuli in the environment both co-localized between the prefrontal cortex and the amygdala. So there's something similar between what the brain is doing at rest and what the brain does in response to emotional events. Over time, what we found was the best predictor of that future resting state connectivity, the one that seems to be most associated with individual differences in mental health, was the stimulus elicited connectivity 2 years earlier. So in other words, the way that we see these data instructing us is that there's this stimulus elicited molding of the connectivity between amygdala and prefrontal cortex that over time, almost in a Hebbian-like way, recapitulated at a systems level, settles into the nature of resting state connectivity that we see at older ages. Now, if we pull that prediction strength out and see how it differs by age, we get the following. So on the y-axis, I'm plotting the strength of that prediction from the stimulus elicited to the resting state connectivity. And we see that that prediction is strongest at the youngest ages and then attenuates as we get older. So in other words, that finding is consistent with the notion that at younger ages, we have a more plastic, malleable system that's more responsive to the environment and is predictive of future resting state. And then that starts to attenuate over time. That prediction attenuates because you start getting more of a bidirectional exchange between these two states of the brain. Okay, so all of that, all of those brain data gets us to this question that really drives our research, which is childhood a sensitive period for the development of the connections between medial prefrontal cortex and amygdala? So we're really compelled by many of the rodent studies and basic neuroscientific labs, finding that early emotional learning is enduring and potent. Often stimuli that are learned early in life can be used later on in adulthood, often to exert anxiolytic effects. So I'd like to demonstrate what I'm talking about through an experiment performed by a colleague, Takao Hench at Harvard. These are adult mice. And if you place adult mice in an open field, they find the center of the field anxiety producing because they're vulnerable in the center. Predators can get them and so on. So they tend to run to the corners as quickly as they can. And a naive adult mouse will typically run to the corner that has a silent nest if Takao Hench's group has placed a music nest in here as well. The naive adult mouse will run to the silent corner. They avoid the music. Unless during the mouse childhood, they were pre-exposed to music. Then in adulthood, they will show a preference for the music nest. Not only do they show the preference, but their preference is specific to the song. So if they were pre-exposed in childhood to Beethoven's Symphony No. 1, then that's what they prefer in adulthood and not jazz, and vice versa. So this learning, this effect is really dependent on this critical period or sensitive period during their childhood. Not only do they form this preference, but in adulthood, when you play them their childhood music, they will show a reduction in anxiety, which is measured here by increased number of times that they start venturing out into the center of the field. And when you look in the brain, green dots here are CFOS data. Green dots indicate more activity in cells within the medial prefrontal cortex. In adulthood, when you play them their childhood music, you get an increase in medial prefrontal cortex compared to music that they were first exposed to at other ages. So we were really interested in this study design for a number of reasons. The primary one being that it's really difficult to think about how do you identify sensitive periods in humans who take so long to grow up. So maybe music gives us an opportunity to probe different developmental periods of life with a little bit more confidence. So we can use music as a vehicle for asking questions about sensitive periods in humans. But instead of classical music or jazz, where it's really hard to know how old somebody was, how old an adult was the first time they heard that music, we used pop music and we used data from the Billboard charts to know how old adults were when they were probably bathing their brains in that stimulus, so that we're really targeting that sweet spot, that putative sensitive period in childhood, rather than some other ages of other periods of development. So to walk you through our design here, if we designed the study in 2012, we had participants come in 2012, and they were young adults. They were 22 years old. So we want to test the efficacy or the sensitive period properties of a stimulus from their childhood. So when they were about 7 years old. So if we do the math, we need a stimulus from about 1997 to present to these young adults. So we searched through the archives, and we found a stimulus that was going to be really helpful for us. So these are the Backstreet Boys was a great stimulus for us. Okay, so to remind you, in the rodent study, they first stressed out the mice. They put them in this open field. In the human study, to stress out our participants, these were UCLA undergrads, and we gave them hard SAT math. And we watched over their shoulders and timed them, and we told them that they were doing pretty well, but a little bit worse than the average UCLA student. And so they should try and increase their accuracy and speed it up a little bit. Then they were presented with breaks. And during their breaks, they were presented with 2 radio stations. One radio station would play songs from their childhood, and the other radio station would play songs from their adolescent period. And the question was, just like in the mouse study, under duress, do we see people gravitating towards one of those stimuli over the other? So if you first pay attention to the green lines, these are the participants who grew up in the US. And what we find is, with increasing stressor bouts, they're increasingly choosing the childhood music over the adolescent music. This is a relative preference here, but if we break this up into people who reported listening to a lot of pop music when they were growing up, this becomes an absolute preference in those individuals. And importantly, this preference, it's a behavioral preference, but it's not reflecting taste. It's not reflecting they're liking the music more. In fact, they're not necessarily liking the Backstreet Boys more than Justin Bieber. But under duress, they have this kind of gravitational pull towards that stimulus. The control participants grew up outside of the US and reported not listening to the Backstreet Boys when they were growing up. So they're more likely to choose the adolescent music. So everyone has their preferences, but it was only the people who were exposed to their childhood songs that also showed significant decreases in galvanic skin response and autonomic response. The more that people chose their childhood songs over the adolescent songs, the calmer they reported feeling. And childhood songs were more effective in increasing activity in medial prefrontal cortex and strengthening those functional connections with amygdala, such that the more that people chose their childhood songs to listen to, and the more that prefrontal cortex activated to those songs, the less anxiety people reported feeling subjectively. So this study gave us a little bit more confidence in this notion that there's something special about the encoding of stimuli that you're exposed to during childhood. But what we're really interested in is not the Backstreet Boys, but we're really interested in the species expected stimulus of our parents. So, when we think about the development of the brain, it's easy to kind of picture it in a test tube almost in a way, and forgetting that it's happening, it's developing in this very special context. And of course, this is a universal for humans. When I talk about the parent in humans, I can be talking about the mother, I can be talking about the father, I can be talking about the adoptive parents or the grandparent, because this relationship is a learned relationship. This attachment is a learned behavior. One thing, and this is true for many altricial species, not just the human, but what is possibly unique about the human is how long we spend in this juvenile state hanging around our parents. which is a curious design because it's metabolically very expensive to raise another human being. And so it begs the question, why would Mother Nature design a species such that we spend such an extended amount of time with our parents? And the argument that we find really compelling is that this buys us the luxury as developing humans of this protracted period of brain plasticity, that it gives us this incredibly long period for learning all of the massive amount of things we need to learn about our emotions, about other emotions, about the environment, and so on, that produces the incredible affective complexity that we observe in adulthood. And so in that way, we can think about caregivers or parents as providing a scaffolding, like on a building, providing a scaffolding around the development of our central nervous system, so that our central nervous system in adulthood should reflect the nature of that scaffolding that was once there. So the rodent data, again, becomes very useful for us here because it's much more advanced in understanding this intimate relationship between amygdala development and the role of the parent. And so I'm sharing with you here data from a colleague, Regina Sullivan at NYU. On the x-axis is rat age. And what her laboratory has found is that early in development, there's this parent-dependent sensitive period for amygdala functioning. That is to say, when the parent is in the nest with the rat pup, the parent keeps amygdala quiet in the rat pup. When the mother, I'm saying the mother because it's the rat, but when the mother is out of the nest, the rat pup's amygdala increases its activity. So the parent's present is literally essentially turning on and turning off, or modulating the activity of the amygdala. Now, to test what effect that has on behavior, her group has used Pavlovian fear conditioning, really the workhorse of amygdala behavior in the laboratory. She pairs a peppermint odor with a mild foot shock, and then places the peppermint odor in one arm of a Y maze. And what happens when you place the rat pup in this Y maze, we would expect them to avoid that peppermint odor, just like you and I would. And the reason you and I would avoid that peppermint odor is because our amygdala is active and mediates that type of avoidance learning. So if the rat pup is fear conditioned without its parent present, then the rat pup does just what I explained. It avoids that peppermint odor because the parent wasn't there, the amygdala is on, so you learn avoidance. However, if the conditioning happens in the presence of the parent, then the animal shows this seemingly bizarre behavior, which is they now are more likely to approach that peppermint odor because the peppermint odor was learned in the parent's presence. The animal, the rat pup, is associating that peppermint odor with cues of the mother. And the mother's presence essentially keeps the amygdala quiet so that the animal does not learn avoidance learning, but instead shows this preference behavior. And this preference behavior, which is only going to show up during the sensitive period of development, is the prerequisite for what we call attachment behaviors. In order for attachment to occur, the amygdala interferes with that. So you want preferences to establish when you're talking about anything related to the parent. So the preference for these things associated with the parent is the foundation of attachment behaviors. And attachment is a survival strategy, perhaps the #1 survival strategy for a young altricial animal. Unlike an adult who often relies on fight or flight strategies, a young animal can't do either very well. So their best bet is to attach to a caregiver, regardless of whether that caregiver has been associated with rewarding stimuli or with aversive stimuli, as is the case here. So the parent here during this window of time is an effective buffer against stress and fear-related neurobiology. Importantly, this effect depends not only on developmental timing, but it also depends on the parent being calm. If the parent instead were distressed or anxious, the parent would not have this buffering effect. Instead, the opposite would happen. She would have a potentiating effect on the young pup's stress reactivity and fear learning. So many human researchers got very interested in this finding and have wondered, given that human development is so stretched out compared to the rats, might we see evidence of parental buffering in childhood? And so these are data from Megan Gunner's lab at the University of Minnesota, and she brought in children and had them prepare for a public speech, which is a wonderful way to elevate the stress hormone cortisol. And what she found was, when children were preparing for their speeches, they either sat next to an unfamiliar experimenter or their parent. When children sat next to the unfamiliar experimenter, they showed this beautiful elevation in cortisol, like you and I would. But when children sat next to their parents while they were preparing, they then buffered this cortisol release. So children were not showing that same cortisol effect if their parent was there during preparation. Interestingly, she repeated this with adolescents who did not show this parent buffering effect. They were mounting a very nice court response regardless of the social condition. Our own lab was interested in this parental buffering effect, but we wanted to revisit this fear learning paradigm that Regina Sullivan's group had developed. And so we paired with her team and we presented to young children, 3, 4, and 5-year-olds, a fear conditioning paradigm. So a blue square co-terminated with an aversive stimulus. Here we didn't use a shock, but instead we used a very aversive noise. It was loud, like loud nails down a chalkboard. So blue square always co-terminated with that terrible noise, and the purple triangle was safe. It didn't end with that noise. So children either learned these associations alone or with their parents seated next to them. Then children were placed into a human Y maze, which was a play tent that had the blue square, the CS plus, on one door and the purple triangle on the other door. And children were asked to enter this Y maze and look at both doors and pick a door, and regardless of which door you pick, you're going to get a prize behind it. And we showed them it was the same bucket of prizes behind both doors. All we really wanted to know was which door were they gravitating towards? Would they show this similar parental effect on children's learning as a function? Yeah, right. Okay. So what we found was when children conditioned alone, they were statistically more likely to do what you and I would do. They would avoid that nasty blue square. However, when children conditioned in the presence of their parents, they were statistically more likely to do just what the rat pups did, which is to approach that nasty blue square. So importantly, these are the same children. This was a within subjects design. So that means the same children's learning was differentially affected depending on whether their parent was present or not. So we see striking parallels between what's found in the rodent data and what we're seeing in young children. And this effect of the parent was attenuated in children with higher cortisol levels, suggesting there are some biological constraints on this learning. We also were curious to know if parents were having a similar buffering effect on the amygdala. So we presented children with pictures of their parents while they were getting scanned and compared amygdala response to pictures when they were looking at pictures of somebody else's parent. And what we found was in childhood, we get a dampening of the amygdala response when children looked at pictures of their own parents, which we interpret as a parental buffering effect during childhood. We didn't see that effect in adolescents whose amygdala was not being as modulated by their parents, which isn't to say that parents aren't important during adolescence. They are, of course, but it may be at the level of the amygdala, the parent has started to lose his or her efficacy in modulating activity. We also found that the parent's picture was effective in strengthening that more adult-like regulatory connectivity between amygdala and prefrontal cortex, again, only during childhood. So taken together, what we think might be happening is that when the parent is not present, when the parent is not present, we see this childlike, non-regulatory connectivity pattern or relationship between amygdala and prefrontal cortex. But when the parent becomes available, they help instantiate this more regulatory pattern with the prefrontal cortex. And although we haven't looked too much at individual differences, we do find that this effect of the parent on connectivity with the prefrontal cortex is stronger for more secure relationships between parents and children. Very quickly, I just want to share with you, we also find that if we can have parents have the opposite effect. So here we fear condition the parent, videotape the parent getting fear conditioned and showed that to children. So the children themselves were never fear conditioned, but the parent was. And then we would show children stimuli that their parents watched versus stimuli that somebody else's parent was conditioned to. And what we find was children could learn to dislike both stimuli, but they learn better to dislike the stimulus that their parent was fear conditioned to. And the stimulus that their parent was fear conditioned to was more effective in increasing amygdala activity in the children. So putting these things together, here's childhood. We see that parents are effective in both upregulating as well as downregulating the stress-related neurobiology. And so we think that this is a sensitive period, or at least our findings are consistent with that idea so far, whereby parents have in many ways privileged access to this limbic circuitry. And we have some ideas about the neurohormonal cascades that are either switched on or off as a function of parents' availability. And the nature of functioning in emotion regulation systems after this period, in adulthood, for example, should reflect the nature of learning that happened during this putative sensitive period. Okay, in my last few remaining slides, I just want to share with you some of our findings in the context of childhood maltreatment, because this framework that I've been describing helps generate some hypotheses about why children who experience early maltreatment are at such increased risk for emotion regulation difficulties. And very briefly, I'll say, although our laboratory has been interested in many different forms of maltreatment, the one that we have paid closest attention to is the form of early institutional care, or often what we call orphanages. And this is a highly stressful experience for children in many ways, largely because this is being raised without the species expected stimulus or mega stimulus of a parent. So we often think about this as an omission of experiences, which it is, but I'm always, quick to say that this omission of a species expected stimulus is not the same thing as an absence of stress. In fact, it may be a more stressful experience than many others that an infant can experience. So this type of experience, as well as other forms of emotional neglect, this absence of a developmentally expected input is unfortunately, a highly pernicious experience. Unlike many physical forms of maltreatment where the signs can be more obvious, these emotional forms tend to be more invisible and therefore often overlooked, but unfortunately very common. It's characterized by the failure to receive that serve and return between parent and child that we know is so critical for healthy development of these emotion regulation systems. And children are often described as raising themselves to a great extent, which is a massive stressor for human brain development. When we consider this a stressor, we can turn to decades of neuroscience research showing us that the amygdala at the cellular level tends to show hypertrophy and hyperreactivity following chronic early stress, which is in stark contrast to the neighboring hippocampus, which tends to show decreases in dendritic complexity and size and function. And the reason that I have these two next to each other is because it's a reminder that stress doesn't seem to have a uniform effect on the brain, but instead leads to differential adaptations in the brain that hopefully are helping the individual manage their particular circumstances as best as possible. When we look at the developmental literature, what many labs increasingly have been finding is that stress early in life, when you look during development, seems to accelerate development of these systems. And this acceleration is understood to be a developmental adaptation to those early stressors, which is an activity-based change in the brain to meet immediate needs. So this is a slightly different view from how our field often thinks about the effects of stress on the brain. Our typical view is that of what people often call a deficit view, that stress leads to a decrease in function. That may be true in some instances, but I think it leads to us often forgetting this adaptation view, that often what we're seeing is the individual maximizing their interactions with the environment through brain development. And that allows us to consider the benefits of those adaptations as well as the costs. So very briefly, the children that we have worked with have experienced early institutional care that is then followed by adoption into families that provide a very nurturing environment, and importantly, families that clearly have a great desire to provide care. This is all through international adoption, and international adoption is a very difficult thing for families to go through. So this is a major stressor for a human infant, and because of that major stressor, children are at risk for a number of developmental struggles. However, following adoption, researchers also find that children rebound in many domains. There's tremendous heterogeneity, which I will point out to you in subsequent slides. But when children struggle, it's very often in the domain of emotion regulation difficulties. So the children that we've been working with overwhelmingly were placed in institutional care at birth up to about 6 months of age, and the majority were adopted by their second birthday. So these early experiences are occurring pre-explicit memory. So if we look broadly at internalizing problems as well as externalizing problems, these are data in 373 children, we see that PI, previously institutionalized individuals, are at significantly elevated scores compared to those with a typical caregiving history. And I'm putting here the Cohen's D, showing it's a large effect size. But if we look at the individual children, we see that there's a large spread. Some children are really struggling, and some children have scores that look like children in the typical caregiving group. And so a lot of our work is aimed at trying to understand not only the source of these individual differences, but also how these individual differences may reflect adaptations that the child has made in response to these early experiences. Now, if we look at amygdala responsivity in response to fear faces, these are data from 2 separate cohorts, one in New York and one in Los Angeles. We find that a history of institutional care is associated with amygdala hyperactivity. And that's consistent with the greater vigilance and more difficulty regulating emotions that are children with this experience are at risk for. But we wanted to know, in following this question of adaptation, are any of these differences that we're seeing actually serving an adaptive value? We presented children with a decision-making task, an exploration versus exploitation task. Essentially, children were asked to pump up a virtual balloon on a computer, and every time they pump, they get more money. But at some point, unbeknownst to them, the balloon could explode, so they would lose all their earnings. So you need to make a decision about whether you're going to continue to explore to pump the balloon, or whether you're going to cash in early, whether you're going to exploit your earnings. And we modified this task such that there were two different colored balloons. Pink balloons would let you keep on pumping. It was very generous. And the blue balloons were harsh. They popped early. But children didn't know this at the beginning of the task. They had to learn these associations. Okay. So what we find is that for the exploratory strategy that I'm going to keep on pumping, that was a strategy that children in the comparison group with the typical caregiving history were more likely to employ. Whereas children with the history of previous institutional care were more likely to rely on the exploitative strategy. So they had different strategies in approaching this task, but importantly, they both ended up with the same earnings at the end of the task. So there were, different roads to Rome, so to speak. And the anxiety, the heightened anxiety that was more characteristic of those with the early life stress was actually informative for us in knowing what strategy they were likely to use. Now, if we look at what it means to rely on this exploitative strategy in the context of that generous pink balloon, we see that it is not advantageous to be exploitative. It's better to be exploratory. However, in the context of that harsh blue balloon, now being exploitative actually has advantages for earnings, and you can see that the children with a history of institutional care with the dark in the dark dots were now outperforming the comparison group. So I just want to show you a couple more slides before I end. We examined the connections between amygdala and prefrontal cortex as a function of a child's early experiences. And we find that these are the data I showed you at the beginning of the talk, where children with a typical caregiving history showed a childlike, non-regulatory positive connectivity pattern, which switched in adolescence to that more regulatory mature pattern. We replicated that in a separate group. But a history of institutional care was associated with the more mature phenotype in the brain during childhood. And so what we think we're seeing is that if this is age here on the x-axis, strong amygdala activity at an earlier age due to early life stress may actually prematurely instantiate the onset of maturing of those adult-like connections. And that may be immediately beneficial for the individual to help manage some of those strong emotions associated with amygdala hyperactivity. However, it may have a cost at the same time in that it's reducing that developmental plasticity that I talked about at the beginning of my talk. So I'll just, because I'm running out of time, I'll just say briefly that we do find that children who show a more adult-like phenotype in the brain with a history of institutional care do seem to benefit in terms of their anxiety relative to their peers with a history of institutional care who show a more typical childlike pattern in the brain. So there does seem to be some immediate benefit for this adaptation in the brain. But to address the question of, is this accelerated development also limiting the parental buffering effects on the amygdala? We repeated that parent-stranger paradigm that I described earlier in the talk, and I'm showing you the data from earlier where children with the typical caregiving history showed that parental buffering. following a history of institutional care, we don't see that buffering on average in the amygdala in response to parents' faces. But we did see large individual differences here, which means some children, even following early institutional care, were showing buffering and some weren't. So we interrogated those over the long term, over longitudinal time. And if you pay attention to the right side of the screen here, what you'll see is children that following early institutional care who did show that buffering of the amygdala at time 1 showed significant decreases in anxiety across a 2-year period. Whereas those children who were more likely to retain their high levels of anxiety were those who did not show that buffering effect at time 1. So just for the sake of time, I'll skip here. These data are consistent with behavioral findings from our lab and many other people's lab as well, that the stronger the security and the relationship between parents and children, the lower the internalizing problems children seem to exhibit. So it may be that strong families are always really important, but they may be playing a particularly important role following exposures to early life stress. So just to conclude, I've said that human amygdala PFC circuitry develops very slowly in humans, but that there may be great value in that slow development. And we get the transition to these adult brain phenotypes starting at the beginning of adolescence, which gives us a very long sensitive period for the environment to influence brain development. And parents are not the only important social inputs, but they may be a primary social regulator of amygdala PFC development. Early adversity does significantly increase the risk for poor mental health, but there are large individual differences that are informative for our understanding of mechanisms of these early experiences. And strong parent-child relationships are incredibly important, especially after early exposures to adversity. And finally, early adversity may be exerting its effects on our development, perhaps through influencing the pacing of our development. So understanding differences that we see in brain or behavior through the lens of adaptation is probably more in line with developmental views of early experiences. So with that, I'll just acknowledge the lab, our funding sources, the families and individuals that participated, and many thanks to you for your attention.
Thank you so much. That was really excellent. So interesting. If people, I think we do have a few minutes for questions. If people can put questions in either the little chat box or the Q&A box, and we can read them out to Dr. Tottenham. Okay, so is there any research on the amygdala activity and emotional regulation skills in the offspring of individuals who underwent early childhood stress, trauma, or neglect?
That's a really great question, and certainly refers to the intergenerational transmission of these stressors. And the lion's share of that work has been done in basic neuroscience models. So for, again, returning to the rat studies, and in part that's the case because rats live much shorter, so you can look at multiple generations at a time, and also the causal inferences are much stronger there. What people have found is that, yes, if this was the work of a former postdoc in my lab, Bridget Callaghan, who's now a wonderful independent scientist at UCLA, if a rat, a male rat, for example, during development experiences early life stress, he will exhibit those outcomes that we would expect. Then he grows up, he mates with a female, and then leaves immediately. So the offspring of that female are never going to meet that father. The offspring will nonetheless exhibit the phenotypes, the stress-related phenotypes that the father had. And it's unclear what the route, what was the means of transmitting that information. It could be related to epigenetic mechanisms. It could be related to differential treatment by the mother. If there's something in her that detects these stress-related behaviors in the father and therefore has a different set of maternal behaviors, etc. So that's the example that I always find most shocking. Most certainly, if the female was mistreated and then has offspring, she is at greater likelihood of passing on those phenotypes as well. The flip side to all of that is that nurturing experiences can also be transmitted intergenerationally. And so in that first study that I described, if the father is provided with enriched inputs after that early life stress, then you are less likely to pass on those stress-related phenotypes intergenerationally. And first of all, that's good news, but it kind of has to work that way, right? Otherwise, we would all be in a lot of trouble if only the bad stuff was passed on to each generation. So I find those data really compelling, but obviously has huge implications for humans. And there are certainly clinically, but also in terms of research, great interest in the parenting of individuals who themselves had less than ideal parenting experiences, because it may be something that influences the next generation. And so I know I'm talking too long, but I'm really interested in this question and this topic. And so a colleague of mine, Mary Dozier, who's at the University of Delaware, has a really, I think, fantastic research program. She's developed an intervention that she calls ABC. It's the Attachment Biobehavioral Catch-up. And it's designed to address risky parenting in order to sort of, disrupt any patterns that had been coded from my own childhood onto the next generation. And what we found is that it's an RCT. That intervention is associated with differential prefrontal cortex development in the 10-year-olds. So the intervention is done early on in parenting, and we see the causal relationship at 10 years later in those children.
Wow. Interesting. All right. We have another. Does early life stress alter just the pace of development of prefrontal cortex top-down mechanisms, with the expectation that eventually development will match that of neurotypical young adults?
Yeah. Good question. Yeah, I wasn't so clear about that. What we don't know. What we think right now, because it's early days, is that by altering the pace of something developing, you also will end up altering the nature of. So it's not just that you've sped up something and now everyone eventually is the same, but there's something about the timing that leads to qualitative differences in the end product. And I often, I often think about cooking analogies, right? If I cook a pie on high compared to the temperature that it's supposed to be cooked, I'm going to end up with a very different product.
And one of the attendees just asked, will slides or articles be made available?
Sure, I can make the slides available to the association if you just tell me how to do that. Any other questions? Okay. Thank you so much for your attention, and I hope you all stay well, and I look forward to the rest of the day.
Thank you so much. That was excellent. The only problem with Zoom is that you don't get to hear rousing applause when you're done talking.